Good afternoon, fellow Rotarians, guests, and friends, and those of you who are on Zoom as well. It is an exciting day to be a Rotarian at the Rotary Club of Cincinnati, Club 17. Welcome. Thank you for being with us. We have a great program for you today. We will begin with the national anthem that will be displayed via video, followed by the invocation and four-way test with Kay Adkins.
Please join me. We are grateful for all that we have, including the challenges we face in our work and personal lives. Our challenges are opportunities to learn and grow in our families and in our community. We are grateful to be a part of Rotary International and of this club for all the opportunities we have to make new friends and to foster service above self in maintaining and improving our club, our community, our region, and internationally. We work together to advance international understanding, goodwill, and peace. We are especially grateful for the fun, joys, and laughter that we share in fellowship. We are grateful for research scientists and those institutions and others who support them in their quest to find better solutions and products that address serious health and wellness problems. We are grateful to physicians, nurses, and all health and wellness workers who listen to the scientists and share their expertise and knowledge to keep us all healthy. Amen. Now let us repeat the four-way test. Is it the truth? Is it fair to all concerned? Will it be beneficial? Will it be beneficial to all concerned? Thank you. That's the first time I've messed up on that one.
That's all right. That's all right. Thank you. You may be seated. Yes. Thank you very much, Kay. Next, we will have our prospective members, guests, and visitors to be introduced by Lisa Blaney-Gazaway, followed by our meeting sponsor of today, which is Western Governors University, and K.L. Allen will bring greetings from our meeting sponsor. Lisa?
Hi. So I'd like all of you to join me in welcoming all of our guests. This is like my favorite part. So first we have Claire Luby Colton. She's the Director of External Relations at Vincent DePaul, and she's Dan Long's guest. Welcome. Next we have Mark Scott. He's the Vice President of Corporate Banking and Commercial Solutions. And he's at First Commonwealth Bank, and he's a guest of Esteban Cali. Welcome. To that, we have Nick Schroth, and he is the Strategic Growth Manager at WGU Ohio, and he is a guest of K.L. Allen. Welcome. Yeah. Next, we have Mike Kelly. Right Path Steps Enterprises, and he's a guest of Melinda's. And last but not least, Ron Hollenbeck. He is with the Centerville Rotary Club, and he's our assistant governor. Welcome. Thank you. Keep inviting guests.
Good afternoon, Club 17. For those who have not had the pleasure to meet, I'm Dr. K.L. Allen, Chancellor of WG Ohio and Regional Director of Central Region. It's great to be back and engaged with Club 17. We're about to show you now our new commercial that we rolled out on actually August the 10th. For those of you that want more information on WGU Ohio, Nick, if you could please stand again. Nick is outside. He's doing some raffles or giveaways, so please feel free to see Nick. And here's our new WGU, the University of U commercial.
College wasn't built for me.
It didn't care if I had to work a double shift.
Or if my kid had a fever.
Deadline's a deadline.
I want to set the deadlines.
I want to take tests when I'm ready.
I want to take courses on my time.
And speed up when I know my stuff.
I want a university that cares about me.
Tests on your time, courses on your time, graduate on your time. WGU, the nonprofit university of you.
Thank you, Lisa, and thank you, KL, for WGU sponsoring our meeting today. Next, we will have a new member introduction that will be done by our director, Molly Riedel.
Thank you, Ms. Melinda. Good morning, Club 17. It's my pleasure today to introduce you to my friend, Ms. Karen Hilton, Hilton, no relationship to the hotel company, which I'm sure she gets asked a lot. Karen was born in Chicago and moved to Cincinnati at the young age of 18 when her father accepted a position here in Cincinnati with General Electric. At the time, they lived in Montgomery. When Karen was 33, she moved with her husband and children to Tampa, Florida, as her then husband had accepted an opportunity with Allstate, and they lived there for 15 years. During that time, Karen was a member of 2 different Rotary clubs in Tampa in different parts of the city and worked at several different banks in the business banking space. Karen wanted to move back to Cincinnati to be near her mother, sister, and friends, so she accepted a position with Fork Bank as a commercial banker just this past April. Fork is spelled F-O-R-C-H-T, but it's pronounced Fork, in case you've ever seen it. They have 3 branches. Karen now lives in East Walnut Hills, and she is the proud mother of 2 daughters, Taylor, who is 22, and Eva, who is 20. Karen looks forward to rejoining Rotary, meeting Club 17 members, and of course, being of service. Please help me welcome Karen Hilton. Yay!
Thank you very much, and welcome, Karen. We have another visiting Rotarian, Scovie Matera. Would you please stand? Welcome. Perhaps a guest of Susan Wilkinson. Thank you for being with us and joining us today. We have several members celebrating birthdays this week. If you are present, please stand so we can recognize you. August 31st, that's passed, Doug Bolton. September 3rd, Carl Kappas. September 5th, Brett Labar. And September 6th, Galen Gordon. Happy birthday to you. The Large Club Conference is coming up. We've been talking about it for a while, and our executive director has been working on it very feverishly. We're looking forward to it. October 13th through the 16th are the days of the Large Club Conference, where we will host large clubs in the United States and Canada. Here in Cincinnati, we're really excited about it. It will be the first time in 60 years. So it's a lot of work and planning and preparation. Some of you have asked, in what ways can I help? What areas do we need help in? We need some airport greeters, and if you're interested in helping in that area, contact Terry Boeing. We also need van shuttle drivers. If you're interested in that, contact David Freeman. We need others to help with Nancy and Jerry Reese in the hospitality suite. And if you're interested in being a greeter in the Hilton, contact Mary and Bob Brandstetter. Those are some areas that we have openings in. Please remember to continue to check the E-Rays. Updated announcements will be in the E-Rays. We are really excited. Our first Women in Rotary event is coming up this year. We're really looking forward to it. It will be an event at Top Golf. We had a great time when we were there before. Are any of you here who were at the Top Golf? Raise your hand high. That was a great time. So we're looking forward to that. Again, we got some lessons too from from Jody Devoyd and from Nancy Reese on golf. So you don't have to be a golfer. It's a great time for fellowship and fun. We have flyers for the Top Golf event outside on the welcome table. So if you have any other questions regarding the event, you can contact Mary Smiley. She is the coordinator of this particular event. So feel free to come, bring a friend. You can invite relatives. This is a great event that we've had before, and we plan to have the same type of event and time this particular meeting on September 24th at Top Golf in Westchester. Members in the news, again, congratulations to Rotarian Nancy Reese. Nancy, stand up. Nancy's just done a great job. Let's give her a round of applause. Nancy has been selected to volunteer at the Ryder Cup golf tournament between the U.S. and Europe. It's contested at Whistling Straits Golf Course in Cola, Wisconsin, September 21st through the 26th. She will be at the VIP entrance, welcoming patrons. Thank you very much, Nancy, and congratulations. Our next week's meeting will feature Cincinnati Bengals Elizabeth Blackburn. She's the Director of Strategy and Engagement. So that's coming up for next week. Before we get into this week's program, we will do Split the Pot. So get your tickets ready. We will ask our speaker, Dr. Ray Takigiku, to pull the winning ticket.
All right, I get the chance to talk to you early. It's a red ticket, and it is 3222. All right.
Thank you, Jody Devoyd. The Split the Pot, anyone can participate in the initial drawing, and then only Club 17 members for the accumulating pot. Oh, she missed it. The accumulating pot was $527. That'll go on into next week. So get your tickets and money ready for a Split the Pot next week. Congratulations for winning the first pot, Jody. Next, we will have the introduction of our program by Steve King.
Hello, friends. Thank you, President Melinda. We have a great program for you today. They say that cancer touches all of us, and I was reminded of that just today. I came into the office, and one of my colleagues, who had been out for the last 3 weeks, was back in her office, and I could see the stress that she had been under. She had been tending to her ailing father, and her father passed away last Friday. So we know that cancer is everywhere, and it's been in all of our lives in one form or another. But it's not every day that you get to hear from one of the researchers who's trying to end that. I'm very excited to introduce today's speaker. Dr. Ray Takigiku is the co-founder, president, and CEO of Bexion Pharmaceuticals right here in Covington, Kentucky. Prior to Bexion, Dr. Takigiku was at Procter Gamble Pharmaceuticals and was also an interim co-director of the Genome Research Institute at the UC College of Medicine. Dr. Takagiku's lifelong research interests have been in the origin, evolution, and sustainability of life. His earliest work focused on elucidating the interplay of oceans and atmosphere and early life forms. Later work encompassed toxicology, pharmaceutical discovery, development, and consumer product innovation. Founding Bexion in 2006, the company now has a novel compound in clinical development for solid tumors, including brain cancer. Dr. Takigiku, I'm going to get it, Ray, I swear. Dr. Takigiku received his undergraduate degrees from University of Colorado in chemistry and microbiology, his PhD in chemistry from Indiana University, and business training from the Kellogg School of Management. Ray and his wife, Margaret Van Gils, live in Covington, Kentucky. Please join me in giving a warm Rotary welcome for Dr. Ray Takigiku.
Thank you, Steve. Thank you, Rotarians, for inviting me. I do speak fast, so I hope to get you out of here with plenty of time for questions. Hotels are not normally confused with my last name, but I am a Virgo, so I'll be celebrating a birthday very soon. So I'm going to talk to you a little bit about, and please feel free, since we're all friends, to ask questions during the presentation. I want to talk to you about cancer writ large. I want to hopefully give you a different perspective on the disease that maybe you haven't heard before, and tell you then a little bit about why we're doing what we're doing at Bexion. So you see up there that the title slide, I mentioned the word lysosome. Don't be afraid of that for those who are non-biologists. I'll talk about all of that as well. So snapshot on cancer. The American Chemical Society every year does a look forward and a look back at expected cases of cancer in the United States, and those numbers don't change very much. I've given you a snapshot, too, of the global situation with cancer. Cancer remains the #1 killer globally. In the US, it's #2, unfortunately superseded at times by heart disease. But those of you who have been affected by cancer or who have loved ones who have been affected with cancer, which I'm sure is everybody here, know what a devastating diagnosis it can be to hear that word cancer. And it does. I don't want to be disappointing. I don't want to be discouraging. I don't want to be a Debbie Downer. But it remains a very serious health issue. And it's a complicated disease. So there aren't really simple solutions here, but I'm hoping that you'll find some hope in some of the new directions, particularly one that we're taking at Bexion. So these are other realities, and you have heard about the rise of precision medicine and targeted therapies. There are folks who kind of take a retrospective look at those drugs that do get approved. This is a paper that came out in 2014 representing those drugs that were approved up to that point. Things have improved a little bit since then. 2014 is not that long ago, but we do have some new therapies that have emerged on the market since then. But if I were to ask you in a vacuum what you thought your longevity would be by taking a cancer drug, what would you say? You would say it would give you another year, it would give you another 2 years, it would give you another 5 years. The reality is that those drugs that were approved with a median of a few months improvement. So it's no wonder that so many people look to alternative therapies. Right? It's no wonder that people believe that there must be a better way out there. There's always a better way. It's true. But we have to be grounded in what we know and what we can understand. If the solutions were that simple, then we'd be all doing it. So why is this true? I'm borrowing from a friend, Dr. Tim Kripe at Nationwide Children's Hospital up in Columbus, and he talks about how cancer mutates. Cancer does mutate. When cancer is present, it's really not a homogeneous group of cells. It's not even all cancer cells. It's a group of cells. So cancer doesn't exist in a mass all by itself. Cancer exists with other cells. They, like all living things, need a community. So they hijack a community of other cells to help them survive. But it's not all one type of cell. So you might be diagnosed with a certain type of lung cancer, and that mass of lung cancer is not all composed of the same kind of cell. It's not identical clones of each other. There's variation from cell to cell to cell. And it's rather shocking to me and a little bit sad that that heterogeneity never was really fully recognized by those of us who are researching cancer until just a few years ago, when a surgeon removed pieces of a kidney cancer from different locations and noticed that depending on where he removed those cells were different. They're different from each other. They're no different than everybody in this room, right? That heterogeneity exists because those cells want to survive. And so a targeted or precision medicine treatment that might affect 90% of those cells is not going to touch 10% of them. And that 10% will go on and mutate further, right? And that is one reason that even though some patients respond to therapy early, they may not respond to it later. So that mutation is real, and that mutation has occurred generally by the time that cancer has been discovered. So it's present. So how are oncologists dealing with it? Today, oncologists are looking forward to using combination therapy, right, to cut off exit routes for those cells that might want to try to avoid whatever treatment is being thrown at them. Unfortunately, combination therapies come with combination toxicities. So again, for those of you who have had cancer or have had loved ones have cancer, you know the effect often that those therapies have left. It's particularly sad that the notion of cancer therapy has usually been to try to kill the cancer before it kills the patient, right? But that is a reality. Those drugs are toxic. They're deliberately toxic. And we try to make them specific to the tumor, but it's not that easy. It's really not that easy. Having said that, how much do we know about cancer today? We know a shitload about cancer. Right? We know so much, at least about one aspect of cancer, and that's genomics. I think most of us are old enough to know when the Human Genome Project first started, right? And it was an ambitious project to eliminate disease. That isn't what happened. We haven't eliminated disease. We've hardly dented most diseases, but we do know a lot about genomes. We know a lot about our DNA. And we know a lot, we probably know most about cancer DNA. So what you're seeing here is a snapshot of what we know, and this knowledge is growing and growing and growing. And there are so many researchers in this field doing so much good work that there's probably today, in 2021, 150,000 publications that will come out, new publications on cancer. 150,000 just this year. For people like me, it's really hard to stay on top of that, right? Fortunately, some journals are better than other journals, so you become selective about what you read. But the point is there's a lot that comes out. And what's come out is that, lo and behold, it's complicated, right? So what we've done is we've categorized certain genomic signatures into families. How many mutations do you think the average solid tumor has? How many genomic variations do you think the average solid tumor has? And those of you who are cancer researchers can't answer this question. Give me a number. 50? Anybody else? Hazard a guess? There's no test here. 10,000. The average tumor has over 10,000 mutations. Which mutation do you target when you're trying to eliminate that cancer? Is there a simple answer? There's no simple answer. So we talk about primary mutations and secondary mutations. We try to, the further up the chain you hit, the more fundamental a biological process you're going to impact, the more likely you'll be creating some untoward toxicity. It's tough. It's very, very hard. But we do know a lot. And we continue to learn more. So I've just kind of summarized a little bit of what I've said on this slide already to you. Something that you may have all heard about recently is checkpoint inhibitors, like PD-1 inhibitors or PD-L1, CTLA-4. There's this renewed hope in using our immune system to attack cancer. It's not a bad idea. It's an old idea. Unfortunately, it took this long for a viable drug to sort of come out to do that. But the notion of these checkpoint inhibitors is that it removes the brakes to the immune system attacking our cancer. So remember I said cancer is not, they aren't cells that live by themselves. They're cells that live in a community, and they co-opt normal cells. So some of the cells that they co-opt, not surprisingly, are immune cells. And they use those immune cells to further propagate themselves even. Those immune cells weren't put in our bodies to do that. They were there to eliminate cancer, and often they do. All of us have cells that mutate regularly, and these immune cells are on guard. Some of them are like Pac-Man running around in our blood system, and they identify those mutant cells, and they take care of it. They get rid of them. It's when it doesn't happen the cancer can take hold. But you know, God and evolution have done a wonderful job of creating our immune system. I mean, think about everything we run into, all of the bad things that we run into every day, and our bodies take care of it, right? So harnessing the immune system is the smart thing to do. And these checkpoint inhibitors have, for some people, been a miracle. But I want to point to some numbers here. The logical thing, right, keeping with this precision medicine idea, is that if you have a PD-1 epitope, if you're expressing PD-1, then a PD-1 drug should help, correct? If your tumor's expressing PD-1. No. PD-1 is obviously involved because PD-1 inhibitor drugs, these checkpoint inhibitor drugs, do work in some people. Turns out it doesn't correlate with the presence of that marker on your tumor. A little bit, a lot of it, doesn't matter. About 15% of the people who get treated with PD-1 for those tumors for which it works will have a pretty remarkable response. That tumor will go away. About 15% more people will have some response. The tumors will shrink. That means for 70% of the people, even if they're expressing a PD-1, the drug will not work. Do we understand that? We don't. We don't. So what have the pharma companies done? They put it in combination with other drugs, right? And they've expanded the utility a little bit. You're increasing toxicity if you do it in unsophisticated ways. But in general, these combinations have not panned out. But because we don't understand it, companies like Merck have, I think, probably 2,000 trials ongoing of combinations with their PD-1 drug. I can lay bets with you that 99% of those won't go anywhere. It's almost a stochastic experiment. Now, will we learn something? We will. I don't know what, but we will. But those combinations haven't really gone anywhere. So there is still a lot to learn. Again, if I asked you in a vacuum, do you think cancer deaths are going up or going down? They're really going up, right? And the kind of explanation for that is that we're a growing population. We're an aging population, especially here in the US. And so the incidence of cancer is going up. Are we saving lives? Yes. Is it better to get a diagnosis? It's never good news to get a diagnosis, but would you rather get a diagnosis now than in 1940? Yes, of course you would. But where do you think these the decline in cancer deaths is coming from? Is it from cancer drugs? It's from us not smoking, right? The biggest change has come from us not smoking. So that's an important lesson here, right? If you don't want cancer, don't invite it. Right? Living a healthier lifestyle is important to avoiding this disease. As I've said to the governors of Ohio and Kentucky, right? I'm in the business of curing cancer, but I'd rather people not get it in the first place. And the stats prove the point. The biggest decline in cancer incidence has come when smoking became discouraged. Now we've got a rise in obesity. And we've got a rise in diabetes. And guess what? We're getting a bump in cancer. So it is important to live a healthy lifestyle. There are different ways to look at cancer, and this is one important way. It's called Hallmarks of Cancer, coined by a couple of researchers, Hanahan and Weinberg, and they talked about cancer having specific sort of characteristics. You can see what they are up there, but all cancers have those characteristics. So that suggests maybe it isn't all about the genome, right? Regardless of what's happening in your DNA, these things are happening. Uncontrolled growth, generation of new blood vessels. Somehow cancer is making all of those things happen no matter where it is, no matter how it's arising. And that's what we're about at Bexion. We're about looking at some of those general features that cancer has and trying to make a dent in that disease by attacking those features. So, I want to introduce you to the lysosome. Everybody heard about the lysosome from their high school biology class. Some learned more about it later on. Most of you probably forgot. The lysosome is an organelle inside of the cell. So cells are not only composed of DNA, right? Cells are composed of a lot of things, just like we are not just composed of a heart. We have lots of other organs, and they're all pretty damn critical. Same thing in the cell. So the lysosome was discovered way back in 1955 when a gentleman named Christian de Duve noticed that a lot of different things in the cell were being shuttled to this lysosome. And the lysosome somehow then became known as the trash bin of the cell or the recycler of the cell, which it does do. It takes care of gunk, and it recycles gunk. But that isn't all that it does. In doing that, it's a kind of orchestrator of cell health. So when disease happens, it affects the lysosome. Right? There are some diseases of the lysosome, but diseases will impact the lysosome. And so when cancer happens, the lysosomes get larger and they get more numerous. And why is that? Well, it's because these cancer cells are proliferating, right? They've got more stuff that they need to get done. So the lysosomes become a little bit bigger, they become a little more productive, they become more numerous, and they become more fragile because they're operating on the edge now. The other thing that lysosomes do is they sequester chemotherapeutic drugs. So when you're getting chemotherapy, those cancer cells are trying to get rid of it, right? Where do they get rid of it? They sequester it in the lysosome. So the lysosome is one place where drug resistance happens early. So is the lysosome a logical target to go after when you're trying to go after cancer? I think it is. Right, the lysosome was such an important discovery that Christian de Duve won a Nobel Prize for it in '74, and people continue to do research in the lysosome. So there's a process called autophagy that some of you know about. It's called self-eating. It happens under starvation conditions. It happens in cancer. Autophagy occurs in the lysosome. And that discovery was important enough that a gentleman named Osumi got a Nobel Prize in 2016 for it. Think about this little organelle has gotten 2 Nobel Prizes already. And yet, who utilizes the lysosome for disease intervention? Do you hear about it? There are people who do it. And I'll talk about why we're doing what we're doing. It's just a pictogram of the lysosome in the cell. One place where the lysosome gets a lot of attention is in something called lysosomal storage diseases. And that's really where our drug was discovered, is in lysosomal storage diseases. And I know all of you do good works for those that are most vulnerable amongst us, the youngest of us, and some people are born with this genetic disorder in which their lysosomes don't work. And the active in BXQ350 is a protein that often goes haywire in a form of lysosomal storage disease called Gaucher's disease. And the inventor of the technology that we licensed from Cincinnati Children's Hospital to start Bexion was a lysosomal storage disease guy. And as he was looking at what this protein, sapsin C, does, he discovered that it had anti-cancer properties. So that was an unexpected and surprising finding, and it was one that we didn't understand. And it's taken a while for us to figure this out, but the good news is it makes logical sense. And the good news is I think we've got an avenue to treat cancer among a number of other diseases because of the importance of the lysosome. So there, I've given you the universe of diseases. Virtually any kind of disease you can think of, the lysosome plays a role in. So now, everything that I'm going to tell you, if you believe it, it's your fault, it's not mine. So I'm going to talk to you a little bit about Bexion. So the active is this protein, sapsin C. It's a small protein, it's twice the size of insulin, and it has some unique characteristics. Among them is that it's attracted to a fat molecule called phosphatidylserine. And the reason that that's important is because we believe that all solid tumors ubiquitously express this fat molecule, phosphatidylserine. So we think that's a universal marker for tumors. Now, it is darn hard to target that specifically, but the good news is that God in nature created a molecule that does it already, and that's sapsin C. And so we think that that is part of the reason that we have such a strong safety and tolerability profile with our drug, BXQ350. The other thing that sapsin C does is it is involved in something called sphingolipid metabolism. I'm not going to get into that, but there are two lipids, ceramide and sphingosine 1-phosphate. And you want to think about them on a seesaw. When ceramide goes up, cells die. When sphingosine 1-phosphate goes up, cells survive. Tumors have a lot of sphingosine 1-phosphate. We want to tip that balance. Guess what makes ceramide? Sapsin C makes ceramide. So we've run phase 1 clinical trials in adults and kids. We've touched about 100 patients, and we've done something called a phase 1, and phase 1 is a safety trial. So in cancer, we can't do that in naive patients. The patients that we got were ones that have exhausted standard of care. The patients that we got were the ones that had no other hope. And we did it across a variety of different tumor types. And the reason we did it that way is because even though what I told you suggests that BXQ may be tumor agnostic, it isn't really going to work that way in people, right? That would be an unrealistic expectation. The expectation would be that some tumors would be more vulnerable than other tumors. And we needed to get some sort of human signal of which tumors we should go after first. And we did get some signals. So those are the tumors that we are going after first. One of them was quite unexpected. Several were unexpected, but we're going after one of them. And the main goal of the phase 1, so these patients in the adult situation, they had undergone 7 prior lines of therapy. So think about how sick these patients are. Think about how desperate these patients are. They failed everything, right? They might have, again, they might have been helped by something, but then they failed, and they failed, and they failed, and they failed. So those are the patients that came into our trial. We did prove that the drug is safe and tolerable. We also saw some interesting evidence of activity. Now, these are the sickest of the sick, so our desire would be to move up in that treatment chain, right? And that's what we're doing. One of the other things that we discovered, speaking to that other lysosome slide that I showed you, is that some of the patients spontaneously reported to us that they had improvement in their chemotherapy-induced peripheral neuropathy. Many of these chemotherapeutic drugs destroy neurons, and that becomes a dose-limiting toxicity. And for some patients, even those that survive the cancer, that toxicity persists, and they have something called chemotherapy-induced peripheral neuropathy. And we saw some evidence that patients benefited from the use of our drug in that direction. Just to show you how important that therapeutic area is, there is no drug on the market for chemotherapy-induced peripheral neuropathy. It is a huge, staggering need, and some of you probably do have peripheral neuropathy, so you know how debilitating the condition can be. So that's an area that we're very excited about getting into. I do speak quickly, but time is running out. One of the areas that people know us as a brain tumor drug, and I'm happy to say we're continuing to work in that area. We've got a phase 2 trial in a rare pediatric brain tumor called DIPG-DMG, which for sad reasons does have some notoriety in the area. It's a rare disease, 300 kids a year get it, uniformly fatal. Nothing touches that disease. So we're hoping that we'll be able to help some of those patients. But one of the signals we got was in colorectal cancer. So that is a big phase 2 trial that, knock on wood, we'll be able to start working on late this year, enrolling patients. I'm going to just let you guys ask any questions at this point in time.
Are there any questions? I know Dr. Takigiku said you could ask questions during his presentation, but his presentation was so succinct there weren't any. But before he closes, he's opened up the floor in case there are a few questions before we end.
Precision medicine has, for example, developed some drugs that are very effective against some specific types, very narrow indications. But if we said lung cancer writ large, right? I mean, there's different kinds of lung cancer. Chemo can extend life a little bit. There are precision drugs like Bruton kinase inhibitors that can actually make the tumors disappear. And so if that mutation does exist, and it's a small subset of lung cancers, for example, then that drug would be great. The problem is, again, this resistance thing happens. So with many of these successful drugs that have worked that way, drug companies have gone on to make second, third, fourth generation drugs. And that's great. You know, for those patients that can benefit from it, they can go to the second generation, third generation, fourth generation. But the challenge of actually eliminating a tumor for good, that's still elusive. Now, I don't want to diminish the importance of the PD-1 inhibitors. Some people respond, very durable response. Yeah, I don't know that there are crossover technologies, but you know, the COVID vaccines, the Moderna and Pfizer ones, for instance, wouldn't have happened without the decades of work on cancer. So those are RNA drugs, and the vast majority of work that has happened has been in cancer, and they pivoted, right? Thanks to Operation Warp Speed, they pivoted to COVID, You know, surprisingly enough to me, and thankfully enough to me, I've gotten the Pfizer vaccine. It works. It works. They're turning their attention back to cancer.
And we have one last question from the Zoom, one of our members on Zoom, Dr. McGilligan. Her question is, are there any current trials open for chemotherapy-induced peripheral neuropathy? I am an internist and have numerous people who would benefit from this.
Yeah, thank you for the question. No, the short answer is no. So what we're doing right now is we'll be looking at newly diagnosed colorectal cancer patients. The standard of care is a chemotherapeutic cocktail called FOLFOX, along with a biologic called bevacizumab. And so we'll be opening up a trial of BXQ plus standard of care versus a cohort of patients on standard of care only. And our hope will be to see some signals in prevention of that chemotherapy-induced peripheral neuropathy, but we've got nothing ongoing specifically for treatment of CIPN, not at least right now, but stay tuned.
Well, thank you very much, Dr. Takigiku. Excellent presentation. We are very glad you came and joined us. On behalf of our efforts with Rotary International and the eradication of polio, based on your presentation today, we will give a donation in your honor. Thank you. And also, we would like to present you with a Club 17 motto pin for this year, Serve to Change Lives. Now we have a special announcement from our Executive Director, Linda Muth.
Thanks, Melinda. The Executive Committee of the Club Board of Directors met yesterday, and at that meeting I announced my retirement. So it's been a pleasure watching our club grow and prosper over almost a decade. And I feel so privileged to have played such a small part in the development of our club into the vibrant and relevant group of professionals that we are today. I will do everything in my power to allow for a smooth transition to the new executive director. I will be staying in my position until approximately December 1st. I know that the Rotary Club of Cincinnati will continue to flourish, especially with the wonderful members, such as yourselves, to do what you do best, and that's serving others. Thank you.
Thank you so much. Thank you so very much, Linda. On behalf, you may be seated just a few more minutes. On behalf of the Board of Directors, I have accepted Linda's retirement notification. We thank you, Linda, Club 17, for an outstanding job that you have done and continue to do. You will be greatly missed. When I thought about, and after I read the retirement letter twice, I began to reflect back on the many areas under Linda's responsibility, of which she does so very well. You know, she is Club 17. I thought about all of the things, and I won't go through them now, but that she's a part of, where she represents us on a national level, on an international level, what she does here for the club, her ability to transition each year to a new president and help us get started on our way, and how she helps each individual member. I think the latest email contact I got from her was 2-something this morning, and that was not atypical. And she sent something else out. We have the large club conference coming up, and that was almost 3 o'clock. She works nonstop. When we come to these meetings, everything is all prepared. We have the speaker, we have our program up here. Many of you don't see a lot of the things in the background that she does, but she does an outstanding and amazing job. Let's give her another round of applause. We love you, Linda. You will always be Club 17. And stay in touch with us. And the good thing is, she's still here. I mean, we've used those terms before with COVID that people don't like to hear, pivot, adjust, and things like that. But we're having to do that, but we respect you, Linda, and when the time comes, like you said, we support you in that, but you're welcome to come back and join us and be with us at any time. With that, about a decade ago, we accepted an offer, a pro bono offer from the Juncker Group to organize and conduct a search for our executive director. That was when Linda came to us. The Juncker Group has graciously, they're members of our club, offered to, on a pro bono basis, conduct and organize the search for our upcoming executive director. And we're really excited about that because they did an outstanding job. I had about 24 hours. I don't know how many people I've been on the phone with. We've been looking at some of the files from before when we had the first executive, well, the last one when Linda came on board, talking with past presidents and members to get ready to put this new search in place. So we're really excited that the Juncker Group has decided on behalf of their efforts to support our club to conduct this search again. Linda is notified, of course, all the board members, and she's met with Deanne and Christy, and I've spoken with both of them as well as we begin to put some pieces together to move forward. You will be hearing about what goes on. We are putting together a search committee. We have just two more responses to. We're moving at an expeditiously rate to make sure that we are able to transition smoothly. As Linda said, she will be with us a few more months. So once we hear back from one or two other people for the committee, it will be listed for you, those members' names in the E-raise. So please continue to check the E-raise. Updates will be made there. And the good thing is Linda's still here, and she's here for some more months. So we're really excited to put some things in order so we can transition as successfully as possible. But no, Linda cannot be replaced, but we will move on with our succession efforts. So thank you so much. That was a difficult announcement, probably the most difficult one since my presidency. But thank you again. Let's give Linda a round of applause. We have some meetings this afternoon. We have the classification committee, which will be meeting in salons F and G on the 4th floor. We have the Cincy Due Days committee, which will be meeting on the 3rd floor in the Julep room. And we have the membership development committee meeting in the boardroom 1 on the mezzanine level. And again, next week, our meeting will be Elizabeth Blackburn, Director of Strategy and Engagement with the Cincinnati Bengals Group. So thank you very much. Go out and make it a great week. Meeting adjourned.